Is there anything people would like to talk about related to old homeworks? Well, the exam. Anybody want to talk about the exam? Anybody got questions? So the exam, as you know, is Thursday. I'm going to expect from you a sheet of paper that is your study guide. That is the sort of formulas and sheets you're going to use during the exam. And you're going to send me such a thing, and I'm going to send you the exam. And the exam is going to be due when you're done. It is supposed to be an in-class exam. It's supposed to take you less than 2 hours. If it takes you more than 2 hours, so be it. But don't kill yourself. Just do your best. Don't kill yourself. And when you're done, send it to me.
This is a little bit of a silly question about the sheet of paper, but just so I don't format it incorrectly. Does it matter, like, just keep it on one side or?
Two sides is fine. 2 sides is fine. A sheet of paper is fine.
All right, cool.
And, you know, don't make it 20 pages. But like if in the end you're desperate to get it to 2.5 pages, and you could shrink the fonts and reduce the margins and make it unreadable without like a magnifying glass, or you could be a normal human being and make it on 2.5 pages. Okay. But the whole idea though is for you to reduce the material in such a way that you've thought about what you wanted. And if you've thought about what you've wanted and you really do want more than fits on a, I don't care. But I expect you're probably not going to want more than about a page worth of material at some level. And again, you know, I'm only having you do that for the simple reason that I believe it's a little paternalistic. It is to encourage you to review the material before the exam, because if you don't, I think you will find the exam surprising, even some of the simpler questions. But if you review it, you'll remember, oh yeah, that isn't so hard. We did that already.
Hi, Dr. Cutler. I just wanted to confirm that we'll be starting the exam all together on Thursday in class.
Probably-ish, whatever. If you email me stuff before class starts, I will probably send you your exam before class starts. If you email me after class starts, I'll send you your exam after class starts. If you don't email me by like 10:30, I'll like start sending you, hey guys, you got an exam. But if it turns out you oversleep or you get up too early and want to start, I'm around, I'll send you the exam. It doesn't matter.
But we will all be getting together on Thursday to work on it in class.
Oh, you mean the Zoom? No, there'll be no Zoom. There'll be no,
okay, that was my question.
Yeah. There'll be no Zoom. No Zoom.
Okay.
Yeah. Thanks. Yeah. No. And God forbid, I don't want to see you work on it. Christ Almighty. No. And I'm not. This is. If we were doing a real class, it would be meeting in room 300, and it would be all together, and you'd be all taking it, and I'd be sitting there pretending to make sure you don't cheat. In reality, I'd be reading my email and hardly ever looking at you, because again, if I do it right, cheating doesn't help. So my goal is to write an exam where cheating isn't a thing. You've either understood the question, in which case it's easy, or you didn't understand the question, in which case you're not going to get it right. And there's just no such thing as cheating. I mean, other than maybe, glancing at your friend's paper. But well, you're all adults. You don't do that kind of crap anymore either. So who cares? Anyway, yeah, I guess don't make a group chat about it, okay? And otherwise it'll all be great. Other questions about the exam?
If we had like certain problems from the homework and we kind of wrote like a framework of how they were solved, is that okay to go on the crib sheet too, or is it just concept?
Anything you want can go on the. So when I took my first year exams as a graduate student, our theoretical ecology professor offered us a sheet of paper, again, to put down whatever we want. And he imagined that we were going to put on that sheet of paper every single formula in theoretical ecology. I was positive I could derive every formula in theoretical ecology on the fly on the exam. So I wrote a sheet of paper that was a bibliography. That was nothing more than important papers, the proper citation on them, and a one-sentence description of what the paper showed. And so I had this extremely long, sort of what amounts to EndNote sheet of paper with almost no formula on it. And then proceeded to produce an exam that was almost a verbatim replica of his lectures. He thought that was really good. So anyway, since I'm the sort of student who used to put stuff on sheets of paper different from what I imagined the professor thought we were going to put on there, I certainly wouldn't object to you doing that. You should put on there whatever you think after studying the material you won't remember versus the stuff you think you will remember. And if the thing you think you won't remember is some complex sort of idea, put that there. If you think what you won't remember is citations, put that there, but there's hardly any in this class. Or if you think the thing you won't remember is a formula, put those there. The other idea, of course, is we shall not examine you on shit you could have memorized. There is no point in determining whether or not you memorized something that you could have. The answer is, I'm sure all of you are capable of memorizing anything I ask you to memorize. So we're not trying to test that. We're trying to test whether you know how to use things that you could easily look up. So put on that sheet of paper things you could easily look up, but don't believe you will remember on the fly during the exam. Does that make sense? And yes, I don't care what you write on there. Like literally, I don't care. It just has to be it should be the things you think will help you the most for the exam. If it's nothing more than affirmations, that's great. Just whatever would be best for you. Other questions? Okay, so when you're done with your little study sheet, send it to me, and I will send you an exam Thursday morning. And when you're done with the exam, send it back. And next Tuesday, we proceed with homework 7, which is the sort of end of one locus natural selection and the beginning of some population structure and 2 locus stuff, I think. We all cool? So today's class, I think technically we're not going to put any of this this material on the exam, but it will be only on the exam to the extent that it uses ideas that we've used, that it echoes ideas we use in class. The ideas may be on the exam, not these papers, but these papers may be examinable on the final exam, let's say. We're going to look at these papers in something like chronologic order, something like the order in which they were written. So we're going to start with the null allele frequencies at allozyme loci in natural populations of Drosophila melanogaster. And we're going to start with Sebastian telling us very, very broadly, what is this paper interested in? What are they trying to understand in this paper? What is the broadest of questions this paper is after?
Okay, they were analyzing the diversity in the sequences of proteins in like for different proteins and different loci in Drosophila melanogaster. And for this, they use the electrophoretic approach to estimate the amino acid diversity.
Well, certainly they do those things, but if you had to sort of summarize in your own words in one sentence, what is this paper trying to understand?
They just want to see what would better explain the excess replacement substitutions in what they're seeing in the protein sequences.
Oh, wait a minute. Are you on the null allele paper with Langley?
Oh, wait a minute. Oh God, I have I have like different papers open here. Just give me a second. I'll use my Kindle because like I have like a whole different thing open in my laptop. Sorry. Just give me a second. I was reading it on my Kindle and just ended up reading a different paper by Chiba Cuadro, and I don't know why.
Did I somehow weirdly assign the
No, just saying I read the papers you're saying. It's just that I ended up like reading another paper by Bigon and Cuadro.
My intent will be to have you read that paper later in the semester, but since we don't know what the word recombination means yet, I didn't want to read Bigon and Cuadro necessarily.
So I opened the one with about the null allele frequencies.
Yeah.
They were just checking the protein sequence diversity among 5 different populations of Drosophila melanogaster, and that's like the best summary I have for that paper.
So if I had to summarize this paper, I would say they're interested in what, if any, evolutionary forces are acting on null alleles. They're interested in trying to understand why null alleles exist. What evolutionary forces, they're interested in the question, why are there null alleles? What explains the existence? How can you why are there null alleles is really, I think, probably the deep question of the paper. And to understand why that is a question, Brooke, what is a null allele?
All right, cool. So to the best of my understanding, basically a null allele, and it's basically a non-functional piece of, or like a non-functional allele. So generally, and they talk about this a little bit with it being like partially expressing or partially functional in some of their results, but generally it's just this version of it that wouldn't be expressing protein. So.
And what is an alleles?
It's a slightly different form of a related enzyme. So like if you were to break it up in a phoresis or something like that, you might actually see they're like just a little bit different in composition, but they're kind of the same. So I think that's why they sort of try to investigate, like, are they functionally different, even though they're kind of almost the same?
Yeah. So an alleles is an enzyme where you have more than one observable band. Right. By the way, this paper. The data for this paper was created, about the time most of your parents were young, so that that the technology that we're using to find proteins involves sort of squashing bugs and running bits of the goo on gels, sometimes staining those gels and looking for multiple bands in particular places when you run them with specific currents for specific lengths of time. With this fancy technology of squished bugs goo run on some sort of gelatin kind of matrix with a little bit of electricity on one end and maybe some stains or not some stains, people are actually able to figure out that there are very specific enzymes, sometimes what those enzymes did, and sometimes that there was variation in those enzymes that they called alleles. And sometimes that variation resulted in an allele that was broken, that doesn't exist, that maybe makes no band at all, or at the very least makes a band that has a protein associated with it that doesn't work. So these alleles are enzymes for the fly that presumably the fly needs, and at least one allele is broken. Is that clear to everybody so far? So,
I have a general question as I was reading about allozymes or alloenzymes or whatever.
Yeah.
Some of the material was saying that like these enzymes are highly conserved, evolutionarily conserved. Is this paper like also sort of making that case? Is that part of that, or was that determined prior to this paper?
It was determined prior to this paper. So, and by conserved, it often means perhaps, I mean, for some of them, we might know the sequence of the allele, perhaps. But more often than not, we simply know that the same enzyme sort of exists in lots and lots of organisms.
Mm-hmm.
Like in a second, we might talk about, we will talk about, say, table 1, and we'll talk about some of the things that are on the left there and what those mean. And some of those little letter combinations are going to turn out to mean genes that are that you have, that I have, that these are enzymes that are more or less conserved among everything that lives, which would suggest that not having it might not be completely compatible with life.
Right.
But we'll get there in a second. But yes, it will turn out that almost everything we knew about in the 1970s were things that were fundamentally essential to life, or at least close to that. There were things that you could find all over the animal kingdom. In part because what you do is you'd find an allozyme in one species and then find the same stain works for it in a different species. And that works for it because more or less the protein is kind of sort of the same in all of these organisms, so that the stain largely works. Questions? So, in fact, I think that's probably a good segue for Marissa to start talking us through table 1. What are all those damn columns and rows and what's going on in table 1? Is Marissa here? No Marissa. Okay, is Carmen here? How about Carmen? You want to talk us through table 1? Can you?
Sure. Let me pull it up. Should I share my screen?
Sure, that'd probably be easiest.
Oh, can you enable that?
Oh, can I enable you? No, of course not. I forgot, as always. There you go.
Okay. So this is a list of all of the autosomal loci that they looked at, and they have the North Carolina population, Great Britain population, and under here it's just the X-linked loci, and in the total one, under that line where it says total, they describe the mean frequencies and the total frequencies they see. So what they saw is that all of the X squared seem to be significant with various degrees of freedom.
So first tell me, before we get to there, before we get to anything like complex math, just tell me what the columns are. What's the first column there?
The number of, like, total number of loci.
Are those loci, or are those, what is that number there?
This number,
Yeah, under n.
the total number of genes that they looked at,
Total number
Or
of flies.
flies that they looked at.
Individuals, yeah. So under n, it might be actually alleles, but I can't remember if it's
Okay.
alleles or it's flies. But it's so it's either it's 2n. I don't actually remember if that is the number of flies or that's the number of alleles. But I think it's the
So
number
the
of flies.
alleles.
It says alleles.
Yeah.
Yeah. And k there, if n is the total number of alleles, what is k?
The observed number
Of what kind of alleles?
of like the null alleles.
That's it. So 3 null alleles out of 782 total alleles. And what is that got to thing there? What is that? That's
I thought it was like a chi-squared.
no, the thing on the far left that says GOT-2 or PG, what do the rows represent?
Oh, okay. So that's the locus name.
That's it. Yep. So along my rows, I have a bunch of genes with names like GOT2, PGK, ADH, PGI. Some of these are famous genes like alcohol dehydrogenase, ADH, aldehyde oxide, I don't know, aldehyde OX, PGM. A lot of these are actually, I think some of these are actually in the citrus acid cycle. But these are all enzymes of some sort. In the first set of columns under N, we have the number of alleles found in North Carolina for this locus. The number of, so this is essentially the number of flies they squished, or half, or like twice that is the number of alleles is twice the number of flies they squished. So it's the number of alleles they were able to count. K is the number that appeared null. And then from a second population in Great Britain, they have the number of flies squished, or the number of null alleles. And then they did a little chi-squared test to ask whether these values were, I think, different than whether there was a difference in these values between North Carolina, I think between North Carolina and Great Britain. But we're not going to make much of that. We don't really sort of care very much. All you can sort of largely see is, for the most part, the number of null alleles in North Carolina and the number of null alleles in Great Britain really aren't that dissimilar from their difference in sample size. To a first approximation, it looks like North Carolina has twice as many flies and North Carolina has twice as many null alleles, more or less. There may be one or two little outliers here, but who cares really? For the most part, we have some number of alleles, some number of nulls, and similarity between the two populations.
Just out of curiosity, I see that their design is like basically to choose geographically isolated populations of flies, because they're basically like saying like, you know, we've crossed this body of water more or less, like Japan, Great Britain, and North Carolina. But I mean, one of the things I was thinking about when I was reading this paper was like, I mean, North Carolina is a pretty large geographic area that's all butted up against other geographic areas. Isn't there some sort of like interbreeding between different flies in those areas? Like, and would that matter at all?
Carmen, would you scroll up to the top of this paper? All the way up to the top. So the first thing to notice is where the paper comes from.
Yeah.
So there's a good chance these flies were collected by someone sticking a pot of bananas outside and letting them rot and finding the flies.
Yeah.
The next thing to know is the third author on this particular paper did his PhD somewhere in Great Britain.
Mhm.
So I think what we have here is a collection of flies from Chuck's bananas and a collection of flies from Andrew's bananas. And did they spend a lot of time thinking exactly how they want? No, my guess is these things are like literally from outside the window.
Okay.
That'd be my guess. But we could track down Chuck or Andrew and ask. But these people are very much population geneticists, not ecologists, and they are not thinking at all at this stage about subtle, sort of ecological differences. They're not thinking about subtle differences between one population and another. They are asking the question on a global scale.
Mhm.
If I pull flies from, like somewhere in Europe, and I pull flies from somewhere outside my office, roughly do I get a different answer?
Yeah.
The answer is not really, maybe, but
Yeah.
but not on a, like, it's not grossly different. If you had done the study only in Great Britain, you would have come to roughly the same conclusion. You'd come to doing it only in America, or if you do them
Right.
all together, you're going to come to the same conclusions.
Right.
So this was almost surely a sample of convenience rather than a sample of ecological importance.
Okay. So I was thinking about it a little bit deeper than the authors were, I guess.
I suspect you are here.
Yeah.
I suspect these are populations that Chuck established at NIH, or Andy or maybe Robert brought with them from England. Okay, all right, thank you. We're going to talk about the bottom of the table in a little bit, but not quite yet. So
Can I ask a question about the table?
yes.
So they have these chi-squared values, and they say that they're comparing them with the expected value you get from a Poisson distribution. But how do they know the frequency to put into the Poisson distribution?
I think they've used the mean frequency over everything.
Oh, from their experiment already?
Yeah. I think they've used
okay.
this at the bottom, the 2.5 times 10 to the minus third, 2.31 times 10 to the minus third. I think they used that.
Oh, okay. To see if they're like all about the same, I guess. Okay.
Yeah. But regardless of I don't want to make much of any statistic of whether or not there is any statistically significant difference in allele frequencies between loci or between Great Britain, other than to say, it's something like, 5 in 1,000 at all the loci, kind of, sort of. It's hard to see a lot of different, like, maybe AL dox has a higher rate of null alleles than any other allozyme there. But the vast majority of them, it's higher than 1 in 1,000, it's less than 1 in 100. There's some, but not so much. And there are no grotesque differences when we look at different populations around the world. There are probably subtle differences. There are surely differences in ecology and evolutionary history between these flies. But on a grotesque scale, you would be very hard pressed to say these things are dramatically different numbers. And you'd probably get yourself in trouble if you tried to make any strong claims about one thing being different than another, really. And that's all we want to get out of this, that across all of these loci, whatever there are, 15, 20 of them, and across, 500 alleles, or maybe even 1,000 alleles, 500 flies from any of these allozymes. The null allele frequency is small, but it's generally, bigger than 1 in 1,000, maybe.
because a lot of their samples, they're only getting like one null allele out of like 700 or something. That seems like a pretty low number. Is there a reason why when you have something like that, you need to do this chi-squared type of thing?
No, no, no. I don't think it's actually an appropriate statistical. I mean, like I would not attempt to defend the statistical analysis. I think to the extent that it is here, it is to say that there is nothing grotesquely, dramatically, disgustingly different most of the time. And I think we can all agree to that. But whether testing it against a Poisson against the global mean is the best thing to do here, I don't actually think that that's probably the most appropriate test here, you know. But it's something. Almost anything you do is going to give you the same answer here. You know, most of them are probably not different. Maybe one or two of them are a little weird. Maybe. Try to account for multiple tests.
I have a small question. Like in terms of the null alleles, would you expect like similar trends among genes belonging to the same families?
Yeah, I mean, in general you would, but here these genes are such different families, I might just now conclude you expect the same thing in all genes. Or at least all genes that were discoverable in the 70s. Maybe it will turn out that there's some significant differences in genes that are a little harder to find because they have a little less impact on the organism, but we'll see. We happy with this part of the table? This is just counts. Counts of alleles, counts of null alleles across several different genes. Now, Linnea, you're actually up next with the bottom of the table. Oh, you're silenced again.
Sorry. I actually didn't download the paper onto my.
Oh, okay. Let me share my screen then. I will.
Thank you.
I think I have. Well, let's find out if I have the paper. Let's see. Share screen. Genetics. Okay. So.
Okay.
Tell me about this bottom half of the table here.
Oh,
Why-
okay. Sorry. Okay. I think you might have cut off a little bit, or maybe I did because my connection's a little bit spotty. But I think you just said talk about the bottom of the table.
Yep, the X-linked, this bit here, it says X-linked loci number of alleles scored.
Okay. So my interpretation of it, it was just the fact that they didn't find any null alleles. So they were just saying like, hey, we didn't find any, but we only tested this many. So it could be less than that or something like that.
Yes. So the
yes, but I didn't really understand why that was the case. They said something a little bit about like the hybrid dysgenesis stuff, which I thought was interesting.
We'll get to why.
Okay.
We'll get to why. But so right, this is the exact same table as above, only for whatever reason, well, for an obvious reason that we'll get to in a second, we have now written our genes in columns rather than rows. We have 1, 2, 3, 4, 5, 6 genes, or 5 genes, rather than 20 genes, or whatever that is. And we've now added another population of flies collected from Japan. Oh, wait a minute, we got collected probably from by Sato. We've added another population of flies collected from Japan to look at the X locus. And instead of reporting the number of null alleles, we've left that off because the number is 0 for every locus in every population. There is no such thing as a null allele found on the X locus in any fly in any population for any of these 5 genes. Is that sort of clear? It is the same table, just instead of repeating the number 0 over and over and over again, they just give you the number n because it's more, it compresses into space easier. And there probably is sort of if you did a statistical test expecting something like 2 in 1,000 alleles to be null, and you sampled, I don't know, what's that, like, that's like 1,500, that's another 1,500, that's another 1,500, that's another 15, this is, this is a number approaching 4,500. And you expected 2.5 per 1,000, you would have expected something like 10 or maybe more of these to have been null. And in fact, 0 were. That number probably is statistically significantly different. There are fewer null alleles on the X chromosome than there are on the autosomes per locus. We will talk about why that is before the paper ends. But that's it. This is, okay. This is 100% of the data for the paper. The data is count of null alleles, count of alleles for autosomal loci, and count of alleles and count of nulls, which is 0 for X. Is there any questions about what the data is? Okay. Mitchell, we are going to analyze that data in a way that attempts to learn something about, evolutionary history and the way natural selection works and stuff. What's the analysis we're going to do?
Is this the that mutation selection balance stuff?
Yeah, that is exactly what that is.
Righteous.
Yeah.
Yeah, so I don't know if I highlighted where they did that.
Maybe. Maybe
And because we did
five.
that, I think in homework 6.
Yeah.
And they were getting values that I didn't understand because they were so small. I don't know if it was like if I had done it wrong on the homework, but they were getting like times 10 to the -3 kind of values.
Yep. You've done nothing wrong. They have done nothing wrong. You are understanding. So the data for the autosomes suggests that the frequency of null alleles is something like 39 divided by 15,000, which is something like 2.5 times 10 to the -3rd. So 2.5 times 10 to the -3rd. And Chuck and colleagues is going to assume that that allele frequency, Q, is in mutation selection balance. So they expect that Q hat is going to equal mu over HS. We don't know what mu is, but the authors assert, from these mutation accumulation experiments that we actually know the mutation rate from these sort of big Mukai kind of cages, that we have an estimate of the mutation rate for at least 6 of these loci that comes out to be something like 10 to the -6th per generation. 3.86 times 10 to the -6 is the mutation rate to null alleles per locus per generation. Something like that is mu. Q is something like 10 to the -3rd. Therefore, HS is something like 10 to the -3rd. So the allele frequency is 10 to the -3rd, mutation rate is 10 to the -6th, HS must be on the order of 10 to the -3rd. That's what we get. And when we use sort of different estimates of allele frequency, different estimates of mutation rates, we can start getting slightly different estimates of HS, but they're still on the order of 10 to the -3rd. We can probably put some kind of confidence intervals on this. It's still going to put HS in the range of 10 to the minus third. So if null alleles are maintained in the population by a balance between mutation and selection, selection against heterozygotes doesn't need to be any stronger than 10 to the minus third. Or if 1 in 1,000 heterozygous flies dies because he has a null enzyme as one of his 2 alleles, that would explain the frequency in the general population. It takes silly small amounts of selection to put even mutations of huge effect, things that fully break the protein, into mutation selection balance, into tiny little frequencies. Could also very reasonably be maintained by selection only against the homozygote, where Q hat was equal to the square root of mu over S. If you think S is around 1, then in mu is around 10 to the minus 6, then you'd expect its frequency to be around 10 to the minus third. So either way, you're going to come up with an estimate that all of this is pretty consistent. A null allele frequency on the order of 10 to the minus third, a mutation rate on the order of 10 to the minus sixth per locus, per generation per locus. That's just mutation selection balance. And every single autosome looked consistent with that story, more or less. Every single population looked consistent with that story, more or less. It's probably just mutation selection balance. So null alleles, things that break proteins, are probably rare, and they're probably at whatever frequency they are because of the specific mutation rates in mutation selection balance, where selection is very, very weak against the heterozygote or is only against the other homozygote. Does this make sense? So that is an estimate of a selection coefficient that is 10 to the minus third. That's how you can estimate a selection coefficient that's 10 to the minus third. That will be very hard for Bruce to estimate. It's the sort of selection coefficient that doesn't easily come out of a population cage or even out of a chemostat. That it's difficult to measure a million of something. Yet, selection coefficients on the order of 10 to the minus third may be what is controlling the frequency of null alleles. Never mind alleles that actually allow for functioning of proteins.
So has anybody squished up enough flies to estimate HS for X-linked? I mean, I imagine if you just did enough of them.
Yep. You will eventually find some null alleles.
Yeah.
But we'll get to that in a second. But the first thing to say is the thing you're looking for is not HS for X-linked, because of those goddamn boys. So whereas we might imagine that in the autosomes, the allele frequency is maintained by selection, by weak selection against heterozygotes, at least it's consistent with a 10 to the minus third HS, so very weak selection against heterozygotes. On the X chromosome, it's almost surely not being maintained by selection against heterozygotes. It's probably being maintained by selection against homozygotes because, well, let's talk about that. So Michelle, first remind everybody of the fundamental differences between boys and girls in chromosome counts.
I don't know about flies.
Flies are humans.
In humans, if you're assigned female at birth, you're probably an XX chromosome, and where males would be XY. So the difference is the number of X's.
That's it. Flies are exactly the same as humans. For all purposes, assume a fly is a human. But at least for this purpose, yes. So flies, female flies are XX, male flies are XY. Meaning that if you are a male fly, you have only one copy of all of your X-linked loci. Meaning if you have a null allele at your X-linked locus, then you don't have a functional copy of that protein. There just isn't one. So Chuck does a little calculation here where he estimates an upper value for the frequency of selection an upper value for the frequency of these unobserved null alleles, but who really cares? The real point here is that the frequency of nulls on the X is much less than the autosomes, almost surely because they're being balanced by selection in male only, in effectively hemizygous, homozygous state. That having no working copies of some of these genes might be lethal, or at least have S's dramatically bigger than 10 to the minus third. So that on the X chromosome, selection is acting probably primarily through its effect on hemizygous males. And these things are really, probably lethals in males, which is why you hardly ever find any. So those are the intuitions to get out of this paper. Null alleles on the autosomes are rare, probably held in check by mutation selection balance. And that selection can be incredibly weak on the heterozygote, 10 to the minus third, and it'll all hold together. On the X chromosome, it's much more likely that selection is actually acting through males, hemizygous males. And as a result, the allele frequency is much lower, because these broken genes are killing boys with only one copy. Does this make sense? This should be your intuition moving forward into whatever organism you are interested in. That null alleles, autosomal null alleles are rare and probably in mutation selection balance, and it might not be much selection that keeps them there. And on a sex chromosome, they are even rarer. And it's actually probably being balanced more by selection in the hemizygous sex. The hemizygous sex is not always male, as most of you guys know.
So I just have a quick question, kind of thinking broadly about how you might tackle that question experimentally. Would you basically then, if you really wanted to understand what might be acting, like would you have to then take a look at gametes basically and look at the frequency of like certain alleles existing in gametes of an organism before like a reproduction of a new organism actually occurs? Or Like,
So if I were doing it,
Hmm?
if I were doing it, I'd probably think about doing that like in something like a pedigree, where I start following alleles through mom and dad to kids, maybe even if I was able to, grandparent to mom to dad to kids. And if I had a few generations, I might be able, in a lot of samples, I might be able to observe that when an allele hits the male generation, it tends to go away or otherwise result in that male not reproducing very much. But when it hits a female, the female tends to sort of survive longer and maybe has kids. And that I might be able to model that. I might be able to sort of see that if I only just had counts of people in pedigrees, I bet I could start thinking about estimating the effects in males versus females, particularly if I made some assumptions. But I think that's probably how I would do it, is try to do it not at a gamete stage, but at a after selection in the organismal stage. But I would surely need some kind of model, and that'd be fit and parameters and stuff. Does that make sense?
yeah, cool.
Okay. That's all I want you to get out of this paper. That one result you were able to prove on the back of an envelope, Q hat equals mu over HS, probably explains everything you need to know about null alleles and just about everything on Earth. That it all hangs together. Alleles of large effect tend to have, it doesn't take much selection in the heterozygote to keep them at a very low frequency, and they are at low frequencies, and they're at even lower frequencies on the X. Right. And again, consistent with Greenberg and Crow, if HS is 10 to the minus third, and we imagine S is probably decent size because these are, again, as we see these allozymes on the X look like they might be lethal-ish, so S might be large. This is all very consistent with when S is large, H is a very small number. And in fact, here, HS is 10 to the minus third. Any questions?
I have a question about kind of the conclusion that they think this mutation selection balance can't be really applied to the whole genome, and their reasoning behind that. And they said something about like a bias of the variability they see for the loci that are actually included in the study. And I was just wondering if you could like talk about that a little bit.
So the first thing to say is they're probably wrong. The next thing to say is in the 1970s, we knew, right? So right now we know about 20,000 genes in most genomes. Maybe the fly genome has 10,000 genes or something. But most organisms, most complex organisms have thousands of genes. In the 1970s, we knew of preciously few, and most of them were intimately related to the production of energy in the organisms in question. They were like citrus acid cycle, they were breaking down of alcohol, there was stuff like that. So the assumption was the genes we knew in the 1970s may be a very biased subset of all genes, and maybe a biased subset in a way that we found them the most important genes for physiologic function. And that once we find all the other genes in the genome, we'll see very different patterns. They at least acknowledge that as a possibility.
Okay.
That possibility will turn out to be largely false. That there are almost no genes in almost any genome that have a lot of null alleles. And when they do have a lot of null alleles, we usually call the gene in question a pseudogene because it turns out it doesn't actually do anything. So it is, in fact, very rare to find null alleles for any functional gene in any organism. They are all general, and when you find them, they are almost always consistent with mutation selection balance. This story, while the authors are being cautious, this story actually explains most large effect alleles in most organisms, full stop. Other questions?
Actually, I'm wondering if this paper can offer evidence for inverse homozygous heterozygous effect.
Yeah, I think it does. I think it exactly does. We see no null alleles in males on the X, suggesting that S is very, very large for null alleles. That's why we're not finding any, that's why we're finding so little of it on the X. So if the selection coefficient is the same on the autosome as it is on the X, S is very large, and we estimated HS to be 10 to the minus third, therefore H must be very small. So these are exactly the sort of alleles that are probably lethal in homozygous form, and H is on the order of 10 to the minus third. This is a perfect example of the inverse relationship between heterozygous and homozygous effects. Other questions?
Can you talk a bit about the transposable elements?
Yeah.
Yeah. Okay.
So do they acknowledge that some of these mutations are due to transposable elements? Do they even know that? They probably.
Yeah, they talk a little bit about it, but they say it doesn't explain it or anything. But yeah.
So TEs were really just coming into evidence of their existence in flies at this period of time. So you guys want a history lesson here?
Okay, maybe maybe not.
So the
doesn't seem too relevant.
Yeah. So the history of transposable elements were always known in corn. That Barbara McClintock showed the existence of transposable elements like in the 1950s in corn, and they were not recognized to exist in any other organism for a very long time, and it was really sort of the 1970s when people started suddenly realizing transposable elements exist in all sorts of organisms, including in melanogaster stocks. And it turned out that the existence of transposable elements in certain melanogaster stocks were the reason some of these stocks wouldn't behave. What happened is we trapped a bunch of transposable elements in them, and then they would just start to die. The longer you kept them in the lab. That was this dysgenesis thing that was going on that they allude to. So it was finally understood in the late 70s that some very, very, very difficult to understand observations in flies were actually the result of transposable elements being in one stock versus another. And the answer is these stocks are not full of transposable elements. So like that's not what's going on. This is not a transposable. These are mutation. This is mutation. This is not transposable element breaking genes. Other questions? All right. Let us stop with the Langley paper and start with the MacDonald-Kreitman paper. So, Kiana, in one sentence, tell me what MacDonald and Kreitman are trying to do in this stupid Nature paper, or the smart Nature paper, or this Nature paper, depending on how you want to look at it.
This paper was essentially a test of the neutral theory of evolution. Essentially, the neutral theory is true, the ratio of
You got it.
I do. The ratio of replacement to synonymous fixed differences should be this. Fixed differences between species should be the same as the ratio of replacement to synonymous polymorphisms within a species. Yes, essentially. And so they just counted for this particular locus the numbers of each and then examined the ratio and found that the neutral theory of evolution doesn't seem to hold in this case.
That's it. That's even more detail. But yes, so the one sentence description is this paper creates a test of neutrality. And the way it implements that test is by comparing the ratio of fixed differences, and we'll talk about what that is in a second, to polymorphic sites. And it looks at those ratios for a few different classes of things and asks whether those ratios are the same or different. And the assumption being things are supposed to be the same under neutrality, and if they are not the same, you can reject neutrality. Okay, that is the one sentence description. And so where do we want to start with this? Holy shit. Let's start. Go ahead. Why not? Mega, let's start with table 1. What on earth is table 1?
Should I just go like column by column, I guess?
Yeah.
So they nicely explained it right below the table, but the first column is all the nucleotide positions. So
let me share my screen so that everybody can be looking at it at the same time. Let's see. Come on. Okay, go ahead. Explain.
Okay, so the first column on the left shows all the nucleotide positions that were looked at. And then they mentioned something about how they used exon 2, exon 3, and exon 4, but I wasn't sure about the significance of that in comparison to the nucleotide positions. I think it was just like saying where they got them. And then the next column was this like consensus nucleotide. And that's kind of what they compared the rest of the nucleotides to. I think it was just for ease. So all these dashes means that it's the same as the consensus, and otherwise it's different. And then there are a few underlined ones in the 5th column. And all the underlined ones are when they had both the consensus and a different nucleotide. And then going through this A, B, C, D, E, F, G, H, I, J, K, L, those letters correspond to different sequences of alleles. So we talked about ADH in the last paper, but they looked at different types of ADH. So there was like ADH, S, F, and then another letter that I forgot.
Fast and slow. We'll get to what that means in a second. That's
okay. And then basically in the last two, I think those were the most important columns. They talked about if it was replaced or not, and then they talked about if it was fixed or polymorphic.
Exactly. So this is a single gene, the ADH gene in flies. And we're looking at, what look like 900 bases, maybe 900 bases of a single gene. And we're doing sequencing. This is not any Alleles nonsense. This is sequencing. This is sequencing before we've really invented PCR. So this is very, very laborious sequence. Marty Kreitman gets his PhD for generating the sequence for the first 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 of these melanogaster alleles. That was his PhD thesis. He generated the first 11. And then it looks like we've got a 12th, and then we've added some simulins alleles, and then we've added some Yakuba alleles. By the way, the phylogeny is melanogaster and simulins are very close together evolutionarily, and Yakuba is an outgroup from those. In fact, the word simulins largely comes from the fact that unless you are good at knowing Drosophila, you can hardly tell the difference between a melanogaster and a simulins. Simulins looks remarkably like a melanogaster. The flies can tell each other apart really easily, but unless you're Dave Begun, you can't, or at least not super easily. These are very subtle differences between the flies. And
Sorry, I have a question.
Yeah.
I'm just curious about the sequencing. Did they use Sanger sequencing?
Technically, they used Maxim Gilbert. So Marty was a PhD student with Dick Lewinton and Wally Gilbert. And so Marty was doing literally Maxim Gilbert sequencing, which involves loads and loads of DNA and water baths, lots and lots of water baths. This is years of work, effectively. After PCR, we get things like automated sequencers. But no, these are out on a gel. 100 bases at a time, 80 bases at a time, that kind of stuff. So we've sequenced like 12 melanogaster, I don't know, what's this, 6 simulins, and like 12 Yakuba. And we're reporting only the positions where there are differences between some of these flies. If this is 900 bases, at something like 850, every single fly, whether he was melanogaster, simulins, or Yakuba, has the same allele, has the same nucleotide. But at 50 or 60 of these, whatever this is, there are differences. At this site, which is at position 781 in the canonical ADH transcript, every melanogaster has a T, every simulins has a G, every Yakuba has a G. It turns out that this position changes the amino acid sequence. The word replacement to a population geneticist means a change in the amino acid sequence. This is a non-synonymous site. This is a site where when you have the T allele, when your G changes to a T or your T changes to a G, the amino acid that gets put there differs. So here, Yakuba and simulins have one amino acid, and melanogaster has a different amino acid. Every single melanogaster they looked at had this different amino acid. So they call that a fixed difference, a difference that between these flies that makes every member of melanogaster different than every member of simulins and Yakuba. Do those words make sense?
I have a question.
Yeah.
Is that the reason why, like for position 816, where it's polymorphic among melanogaster, but it seems to be a fixed difference between simulins and Yakuba, that they still consider that to be polymorphism?
It is polymorphic in melanogaster? It is a fixed difference to simulants and you know, a fixed difference to Yakuba between simulants and Yakuba.
But they still categorize that as polymorphic.
Polymorphic within melanogaster.
Oh, okay. Okay.
And I think we will find that these polymorphic shouldn't say within what species, but I think they're going to call it polymorphic if it's polymorphic in any species. We'll think about that. We'll see how they write down these tables in a minute. We'll see what they do with this. But, oops. So, getting to that, well, first let's look at the next line. Again, we have a fixed difference because all of simulants and all of melanogaster have one allele. Yakuba has a different allele. So this is a fixed difference between the ancestor of melanogaster and simulants and Yakuba. But this site does not change the amino acid sequence. You get the same amino acid whether you have a T here or whether you have a C here. So we say this site is synonymous. Changing between a T and a C here does not change the amino acid sequence. This is a synonymous site. Next one is also a fixed difference. It causes a change in the amino acid sequence, its replacement. We get down here and we find here the T allele is segregating in melanogaster. So we would say, so we have two words for this. We could say the site is segregating, or we could say the site is polymorphic. In either case, it means in the individuals within this population, there are two different alleles present. Is this all clear? Welcome to an era when we published all the raw data. This is the entirety of the data that is going to go into this paper. All of it. Questions? So, let's go with Kevin.
Mm-hmm.
And tell me about table 2.
So in this table, they basically enumerated the polymorphic, replacement, and synonymous substitutions and the fixed replacement synonymous substitutions, and they use this as part of like what they call a G-test, which I looked up, which is actually just a proportional test, looking at proportions. And they mentioned that I think in like the third or fourth paragraph of this paper, they said about 29% of the fixed differences between species are replacement substitutions, but only 5% of polymorphism are replacement substitutions. So they basically use this to like create a proportion.
Yeah. So the first thing to say is if it weren't 1991, where like these guys have actually probably never heard of a computer, we would have done a Fisher's exact test.
Yep.
Fisher's exact test is much more appropriate for sort of samples of this size, but it was God almighty a pain to calculate back in the days when you had to use a pencil. So very few people used Fisher's exact tests before computers. But you and I would use a Fisher's exact test on this. So who cares what they actually did? But we should be doing a Fisher's exact test here. But let's get to the assertions. So they're going to assert that the ratio of, say, fixed to polymorphic replacements ought to resemble the ratio of synonymous fixed to polymorphic things. Mm-hmm. Or the rate of fixed things in replacement over synonymous ought to be the same as the ratio of polymorph. Do we know why this is? Do you know how many synonymous sites, would you have expected to find in this thing? And how many replacement sites would you have expected to find?
My guess is that, I mean, they would be in equal ratios, but I was actually thinking about this as I was reading it, whether they would both be very small. I think.
So if I told you that you sequenced, say, 12 melanogaster alleles and found, actually, let's do it. Let's do it for the polymorphic stuff. If I told you you sequenced 12 melanogaster alleles and you found 2 sites, could you estimate theta?
It'd be pretty hard, I would guess. So,
well, I might imagine that if you look back at your homework, you'd say theta equals the number of sites divided by the sum 1 over I from I equal 1 to n minus 1. It's a simple formula of theta over a function of your sample size. And theta is equal to 4 times the population size times the mutation rate.
Yep.
So the number of polymorphic sites I expect looks like 4 times the population size times the mutation rate over some complex formula of the sample size. The number of synonymous sites I expect is just a different theta. The sample size is the same, and the population size is the same. So the ratio of these two things looks like the ratio of the mutation rate to replacement sites over the mutation rate to synonymous sites. It's 4n mu over some function of sample size, 4n a different mu over some function of sample size. The sample sizes are the same. The population sizes are the same. So the ratio of this to that is just the ratio of the mutation rate to replacement sites over the mutation rate to synonymous sites.
Yep.
And for fixed differences, I expect the number of fixed differences between 2 organisms to look like the mutation rate times the time between them. Here, I expect it to be the mutation rate times the time between them. The time between these organisms, whatever they are, is the same for silent and replacement sites. So this ratio looks like the ratio of the mutation rate to replacement sites to the mutation rate to synonymous sites. And this looks like this ratio is the ratio of replacement mutation to silent mutation. So this ratio here really ought to be the same as that ratio there. Or done the other way around, this ratio here ought to be this ratio here. All of these ratios are really just ratios of mutation rates under the neutral theory. So the test is going to ask whether these ratios are the same, because the only way in which they ought to differ, I mean, these things ought to just all be mutation rate. Mutation rate to replacement, mutation rate to silent, mutation rate to replacement, mutation rate to silent. Does that make sense? So this is the test I'm going to do. I'm going to ask whether these ratios are the same. I'm going to do a Fisher's exact test and get a p-value that's very, very small. When that happens, my conclusion is these ratios are not the same, meaning something more than natural selection, something more than the neutral evolution is going on here. So if I have a significant MacDonald-Kreitman test, I have rejected neutrality. I've rejected that the ratio of polymorphic to fixed differences is the same at silent and replacement sites. Are there questions about this?
I have a question just about this G test that they use. It's like a test of independence, like that. It's basically saying like, like these mutations occur, or the mutation rate is independent no matter if it's fixed or polymorphic. Are the assumptions in line with like, I didn't really look this up, so I'm just asking off the cuff pretty much.
The G test is just it's literally asking whether the ratios are the same. Whether 7/2 equals 17/42.
Yep.
That's all it's asking.
Okay.
And so I don't know how I did that. Okay, anyway. So this is sort of obvious to you. 7/2 does not look like 17/42. Those numbers are very different. Or the other way around, 7/17, that's a number like, I don't know, like between a half and a third. 2/42 is a number like 1/20. These numbers are very different from one another. So even with only 20 or 30, 50 sites, we can get a p-value that's less than 0.1. These ratios aren't the same. And whether we did a G test, whether we did a chi-square test, whether we did a Fisher's exact test, the p-value would be very, very slightly different each time, but they would still all tell you the same thing. 7/2 does not equal 17/42. 3.5 does not equal a third, even at a sample size of 50. Okay, that's good. So that's what we learned, was that 3.5 does not equal a third, and they should be the same. These ratios really should be the same if nothing is going on other than neutrality. So at least some of these values are not at their neutral expectation. And now becomes, the MacDonald-Kreitman paper could have been brilliant if it had stopped right there. These ratios are not consistent with neutrality. We can reject neutrality. The ADH locus is under some form of natural selection. End story. But no one ever ends it there. People generally then start interpreting this in all sorts of weird and difficult and crazy ways. Like, one way it sometimes gets interpreted is people say, let's pretend synonymous sites are for sure neutral. Let's pretend we knew that. If we knew synonymous sites were neutral, the ratio of fixed differences to polymorphic differences is like 1:3 here. The ratio of replacement to polymorphic differences is the other way around, 3:1. That probably means there are too many fixed differences or not enough polymorphic differences. I'm going to pretend I know it's too many fixed differences. Therefore, selection has been acting to create new versions of the ADH allele, fixing them in flies, and that's why this number is so large, rather than say this number is unusually small. Or rather than selection could be acting on these numbers, or rather than say some recombination here and you've got your variance term wrong, or you've really got your variance term wrong here. There are a lot of other possible interpretations. But what we have rejected is that these ratios are the same. And if by magic, we knew what some of them were supposed to be. We could start telling you which ones were bigger than they were supposed to be and which were smaller. But that generally requires very explicit assumptions, almost all of which are going to be wrong. And while there are many of your colleagues who spend a lot of time in literature interpreting these differences, I don't think we've really learned anything from those interpretations. Other than it ain't neutral. That's simple.
Can I ask about their possible explanation being the difference in ancestral small populations and when they say recently expanded populations? I didn't really understand that argument.
Yeah. All right. So hold on. We're holding on to figure out what paper have I supposedly assigned you to read in the second half of this term. I'm not having you read Hiroshi's paper. Okay. We're going to have to talk about this then. All right. So there are some years I assign whole other papers to talk about this. So we don't yet know this, I think. But I don't think we quite know this. I think we're still a little bit behind in the reading. The amount of polymorphism in a non-neutral population is a function of the population size times S, the selection coefficient, or really probably times HS. But we're going to assume always that for this stuff that H is a half. So we're just going to think of it as a function of the population size times the selection coefficient. So under a neutral model, the amount of polymorphism looks like 4 times the population size times the mutation rate. In the presence of natural selection, the amount of polymorphism tends to look like 4 is a function of 4 times the population size times the selection coefficient, 4 in S. The rate of fixed differences is also a function of 4ns. And if 4ns gets bigger than even a little bit, the rate of fixed differences goes to 0 very rapidly. So it is a fact. These people knew it, and it's known even better now, that melanogaster experienced a severe population bottleneck around 40,000, maybe 50,000 years ago. That bottleneck was associated with melanogaster following humans around the world. That humans sort of came out of Africa at some point, 100,000 years ago, 10,000 years ago, sometime in between there. And melanogaster followed them. And so melanogaster's historical effective population size tends to be smaller than a lot of other flies. As a result, NS is a smaller number in melanogaster than it is in other flies, meaning they tend to have more fixed differences and more polymorphism. So once you start accounting for differences in historical population sizes, it's even harder to wrap your head around what these numbers mean. And they're vaguely waving their hands at this, knowing that population size hasn't been the same. Therefore, the expected amount of, under the alternative models, therefore the expected amount of both polymorphic and synonymous, replacement and synonymous, polymorphic and fixed differences ought to differ from one another. This is not explained in any kind of way that makes any sort of quite sense, or that you can wrap your head around. A prediction is a fact, and it's something that Hiroshi Akashi fixes as part of his PhD. He'll go through, as part of his PhD, he'll go through and measure selection on synonymous sites, and then he'll also go through and talk about what sort of changes in population size and selection coefficients are sort of consistent with various results from the MacDonald-Kreitman test. And it's a sort of series of about 3 or 4 papers that sort of illuminate most of these concepts. But the answer is, once you admit selection can be on synonymous sites, once you admit population sizes can change over time, once you admit that under the alternative of selection and changing population sizes, predicting any of these ratios is really, really hard, and it makes interpretation of this a mess. The answer is we've rejected neutrality. And if you can start telling me stuff about population size changes from some external data, or you can start telling me stuff about selection coefficients on some of these sites, I can maybe start bounding others. But it's still an ugly, ugly process. Other questions? That's the MacDonald-Kreitman test. It's one of the first really good tests of neutrality. Second, third, fourth, something like that. HKA is a better test, but the math is harder.
Can I ask a question about the mutation rates they use for non-synonymous and synonymous?
Yeah.
So on the first page, they said they estimate all of them are mu over 3. I wasn't really sure why they did that.
Don't need to. Doesn't care. Doesn't matter. It's utterly irrelevant. Whatever the mutation rate is to replacement sites, whatever the mutation rate is to synonymous sites, the number of fixed differences will be proportional to that mutation rate times time, and that mutation rate times time. So if you take this over that, the times cancel. It's just the mutation rate to replacement over the mutation rate to synonymous. I don't care what those values were. Same thing with polymorphic. This is theta. Over here is this is theta with mutation rate to replacement. This is theta with replacement with mutation rate to synonymous. The ends cancel. That little function of sample size cancels, and they just end up with a mu to replacement and mu to silent. Who cares what those values were? This ratio should equal that ratio, no matter what the values are.
Can I ask a follow-up question really quick?
Yep.
So from reading Gillespie, I got the impression, at least, maybe I am wrong, but that the non-synonymous mutations, I guess, with like no prior knowledge, you would assume that most mutations would be non-synonymous.
Yeah.
Is that the case? Like, is there an estimate for the value on that, or?
Yeah. So right, and it's so dumb that you can't that you can hardly so you probably know from your like almost high school biology class that a codon is 3 nucleotides, that there are 20 possible amino acids. So there are 64 possible codons and 20 possible amino acids, at least if you're in mammals. And so many of those codons code for the same amino acid. And you also probably learned in high school biology that, so you have these 3 positions that most of the time when you change the first position, it changes what the amino acid makes. So a change in the first position tends to be replacement. A change in the second position is like a replacement all the time, just about. But a change in the third position often, maybe even usually, results in the amino acid being the same, being synonymous. So the biology just turned out that changes in the, well, it didn't turn out that, but the biology is that a change in the first position is usually replacement. A change in the second position is like always replacement. A change in the third position is very rarely replacement. So if you were changing DNA at random without respect to what it did, you'd expect 2/3 of changes to be replacement, 1/3 to be silent. And you will get something like that no matter how detailed and complicated a model you make. Even if you start accounting for the fact that CT type mutations are more common than GA, and you will always end up in a world, no matter how, even if you think this is just a dinucleotide, you'll end up with a world where most random changes result in replacement, and the ratio is probably 2-to-1 or 3-to-1 for random changes being replacement over silent. Whereas in almost any data set you see that comes from nature, you will see something like this. The number of synonymous sites within any particular organism, within any particular gene. Like you pick an organism, you pick a guy, you pick me, and you sequence one gene. I probably have as many heterozygous synonymous sites as I have heterozygous replacement sites. I might even have more heterozygous synonymous sites than replacement sites. I do that for flies. I do that for your favorite organism. Most of the time, things in nature have as many or more synonymous sites than replacement sites. The natural interpretation, of course, is that synonymous sites probably don't cause as much selection as replacement sites do, and that changing the protein's primary sequence may on average be bad. Other questions?
Just out of curiosity for the literature, is there any rebuttals by the folks that come up with neutral theory to things like this?
No.
Nope.
Remember, there's the rebuttal is more subtle, right?
Mm-hmm.
Even Kimura never said there was no such thing as natural selection.
Yeah.
What he said is that natural selection's effect on polymorphism is weak enough that we can ignore it.
Mm-hmm. Yep.
So a Kimura interpretation of this is that I told you natural selection was weak and that it has almost no effect on polymorphism, but it does have a significant effect on fixed differences.
Mm-hmm.
What you're seeing is how selection has acted over time on replacement sites. It's very, very rare. Like I'm looking at 1,000 sites here, and there's only like 7-ish that have had any kind of fixed difference. Okay.
Mm-hmm.
That's, less than 1% of sites are being influenced by selection.
Yep.
That's what that's sort of argument Kimura would make, with that these differences that I'm seeing are very, very small and are consistent with the idea that they're explaining only a tiny fraction of all variation.
Mm-hmm. Yep, makes sense.
Other questions?
So I know that they were kind of different papers, but essentially that first paper though sort of pushes back a little bit against, I think, what that argument was, right? Because you have a situation where you have this selection happening. Hold on. I had the thought, but I don't really have the words for it. So, but they just kind of seem like this this first paper is pushing back against the argument that you just mentioned.
The first paper is measuring the selection coefficient against null alleles. But again, like the neutrals would say, null alleles, they have they are evolutionarily dead. No one fixes a null allele. It arises, it sits in mutation selection balance, it goes away. That is also consistent with the idea that anything at high frequency is neutral, which was kind of Kimura's sort of insight view, that if it rose to anything resembling an intermediate frequency, it was neutral. That you can have all this little crap mutation that's bad for you. He'd accepted that. But he would have said that had no role in divergence between species and doesn't really account for much of the observable polymorphism because it's so rare. But Hiroshi Akashi is going to measure the selection coefficients on synonymous sites. And selection is kind of observable everywhere you look. Turns out, DNA matters, right? Organism fitness is a function of DNA, and a whole lot of it matters. Maybe all of it, the vast majority of it at least a little bit. Questions? Okay. So, homeworks 1 to 6 are on the exam. When you are ready with your little cheat sheet, send it to me. I'll send you back an exam Thursday. Try not to spend more than a few hours on the exam when you send it back. There is no time limit. If it takes you into Friday, if it takes you into Saturday, if it takes you into Sunday, I will not complain. But don't do it. Don't fight it. Don't hurt yourself. Just send it back when you're done. But if something comes up, like you got like you're moving, and okay, it takes you 4 days. It takes you 4 days. That's okay. But try to send it back sooner if you can.
Word to the wise, don't move.
Yeah. Yeah. Even if your girlfriend's on the other side of the country.
Yeah, exactly.
All right. If you guys find yourself with questions between now and Thursday, shoot me an email. I will probably do my best to answer them, although I might give up sometime Wednesday night. So middle of the night, Wednesday night emails may not end up answered. Sorry. See you Thursday. We're not going to do class. Just email me. We'll virtually see each other.
Thank you.